Hi, welcome back to all. So today we are going to discuss about the question answer on the biotechnology principle and process. We discuss some of 15 to 20 questions in today's session. You have a 1 minute to solve this question. Before that, I'm introduce myself. My name is Yudhishthir and I take today's class uh, for you. Okay, so I'm launching poll for the technical check. Please answer me. Is my voice clear and the screen is visible or not? I think my voice is clear and screen is visible also. So let's start with the first question. So first question on your screen. The question is the cutting of DNA at a specific location is possible with discovery of मतलब किसके डिस्कवरी के वजह से मतलब क्या हमने डिस्कवर किया ह्यूमन ने क्या डिस्कवर किया कि जिसके वजह से हमें DNA एक स्पेसिफिक लोकेशन से फ्रॉम अ स्पेसिफिक लोकेशन वी कैन कट द DNA and options A. Options are option A is the restriction enzyme, option B is the probe, option C is the selectable marker, and option D is the ligases. You have a 1 minute to solve this question. I'm launching the poll. Please answer fast, then we will discuss the question. 1 minute. Okay, answer no. Okay, I'm in this poll. And question is the cutting of DNA. DNA का cutting हम किस चीज से करते हैं? Directly thing on the specific location, and the cutting of DNA happen with the help of restriction enzymes, and that's why option A is the correct option here. Then what about the probes? Probes का मतलब क्या होगा? So probes are the single stranded DNA. They are single stranded DNA, okay, which are the labeled, okay, and this probe is important to identify the we can say unknown structure or unknown strand of DNA. Okay, next about the selectable marker. So I think you already know about the selectable marker. Antibiotics resistant genes are the selectable markers which present in the plasmid. Next is the ligases. So ligases are the glue, okay, which we can say combine the DNA, or we can say they stick two segments of DNA to each other. Okay, and that's why option A is the correct answer here. Next question, question 2. The introduction of tDNA. So they ask about the tDNA into the plants, into the plants involved. मतलब tDNA जो है ठीक है प्लांट्स के अंदर जिसको यूज करते हैं उसमें क्या इन्वॉल्व होता है एंड ऑप्शन ए इन्फेक्शन ऑफ प्लांट बाय द एग्रोबैक्टीरिया ट्यूबनिफेसियम, ऑप्शन बी अल्टरिंग द पीएच ऑफ सॉइल हीट शॉकिंग the plants, option C exposing the plant to the cold for a brief period, option D allowing the plant roots to stand in the water. You have a 1 minute to solve this question. Please answer fast, then we go go toward the explanation of the ऑप्शंस और क्वेश्चन. ठीक है. सो आपके पास 1 मिनट है, 1 मिनट में उसको सॉल्व करो, जल्दी जल्दी सॉल्व करो. क्वेश्चन को पढ़ो. ठीक है. यू नीड टू रीड क्वेश्चन प्रॉपरली, यू नीड टू रीड ऑप्शंस प्रॉपरली, एंड देन उसके बाद इसका आंसर डिसाइड करो. People who join from the YouTube live, they can answer in the live chat. You have a 30 seconds to solve this question. Please answer fast. Good evening, JNV Vishakhapatnam. Okay, so I'm in this poll, and this is your answer. Here, maximum people say option A. देखते हैं option A is the correct option or not. So the introduction of tDNA. Okay, so here T means T come from the tumefacian. ये चीज को ध्यान में रखना हमेशा. Tumefacian. ठीक है. And this bacteria, the complete name of bacteria is known as the Agrobacterium tumefacium. Tumefacium. So Agrobacterium tumefacium have a ability to, we can say, insertional ability. That means it have a ability to insert their genome in the cell of plant. Okay, and that's why option A is the correct option here. Agrobacterium tumefacium is responsible for the plant cancer. Okay, plant cancer in the sense, actually cancer not happen in the plant, but plant cancer in the sense some of the mass after infection, like when infection happen by the Agrobacterium tumefacium, some of the mass generated or developed on the plant trunk. Okay, and that mass in the general word we known as it is the overgrowth of cell, and that's why I'm said ki it is the cancer like. So Agrobacterium tumefacium is the ability to insert their genome inside the plant cell, and that's why option A is the correct. Infection of plant by the Agrobacterium tumefacium, and tDNA is used as a we can say vector for the plant cell. Next one is the option B. बाकी के भी option देख लेते हैं. Altering the pH of the soil. No, this is the completely wrong. pH of soil ka and tDNA there have no any relationship. Next is exposing plant to cold for the brief period. No, this is also not an option. Next, allowing the plant root to stand water. So this also not option. That's why option A is the correct one. Next question on your screen is, which vector can clone only a small fragment of DNA? मतलब कौन सा वेक्टर नीचे दिए गए ऑप्शन में से कौन सा वेक्टर है जो क्या करेगा, एक स्मॉल फ्रैगमेंट ऑफ डीएनए के लिए काम में आएगा या उसको वो लेके जा सकता है. So first option is the bacterial artificial chromosome. It also known as the BAC. Okay. Next one is the yeast artificial chromosome. It known as the YAC. Next one is the plasmid. Next one is the cosmid. Okay. So I am launching poll for it. Please answer first. You have 1 minute to solve this question. Then we move toward the next portion. People who join from the YouTube live, they can answer in the chat box of the YouTube. You have 30 seconds to answer this question. Please answer fast. यूट्यूब वाले लोग यूट्यूब लाइव चैट में आंसर करो। मैं देख रहा हूँ लाइव चैट, कोई आंसर नहीं कर रहा है। प्लीज आंसर फास्ट। ठीक है, आंसर रॉन्ग रहेगा तो चलेगा। ठीक है, so I end this poll and this is your result. Here maximum people choose option C and option C is the correct option. Then bacterial artificial chromosome have a capacity up to 1000 kilo base pair DNA. That means it clone up to 1000 kb DNA. Okay. Next, about the yeast artificial chromosome. Yeast artificial chromosome have a capacity to 200 to 500 kb. Next one is the cosmid. After that, we discuss about plasmid because plasmid is the correct answer. So cosmid have a ability. Cosmid का कितना ability रहेगा? 50 to 100 kb. So I'm right here now. 50 to 100 kb only. And next about the plasmid. Plasmid have a 15 kb only. And that's why option C is the correct one. So small fragment of DNA cloned by the plasmid. Okay, or plasmid act as a vector. Okay, for the small fragment of DNA. So this is about the plasmid. Coming to the next question. Next question on your screens. Question is a very simple. ठीक है, थोड़ा सा दिमाग लगाने की जरूरत है. So question is, which of following is not correctly matched? They ask about the not correctly matched. ठीक है, so you need to read question properly first. Then for the organism and its cell wall degrading enzyme, they ask in the next portion of question cell wall degrading enzyme. So you need to find cell wall degrading enzyme here. So option A, plant cell cellulase. Option B, algae, methylase. Option C, fungi, chitinase. Option D, bacteria, lysozymes. So I'm launching poll for it. You have a 60 seconds to solve this question. Then we will go into the explanation part. Okay. Please answer fast. You have 30 seconds only. चलो जल्दी जल्दी आंसर करो. Okay, I'm in this poll, and here most of people given answer 1 minute as option B, and option B is the correct option. Then which of following is not correctly match? Correctly match कौन सा है वो हमें देखना है. And in second part they ask about the cell wall degrading enzyme. Okay, so we already know plant cell. The cell wall of plant cell made by the cellulose. Okay, actually this is the cellulase ना, ये cellulose नहीं है, ध्यान में रखना. ठीक है, so cellulose, and for the degradation of cellulose हमें कौन सा enzyme चाहिए? Cellulase. That's why option A is the or we can say plant cell and cellulase is the correct pair. Next one is the fungi. So fungi का जो cell wall made from the chitin. ठीक है, chitin sugar. So chitin sugar के degradation के लिए which thing is responsible? Chitinase. That's why option C also the pair is correct. Next one is the bacteria. Bacteria, you already know we have a lysozymes in the our cell. Okay, and which important for the degradation process. So lysozyme is the enzyme which degrade the bacterial cell wall. That's why option D or we can say pair in the option D is correct. But in case of algae, they given here the methylates. Actually, methylates not important to degrade, not important for the degradation of algal cell wall, because algal cell wall also made by the cellulose, cellulose part. सेलुलोज से ही बना हुआ है algal cell wall, and that's why it need cellulase. इसको किसकी जरूरत है? सेलुलेज की जरूरत है, and that's why option B is the correct option here, which is not correctly matched. Next, DNA fragment generated by the restriction endonucleases in the chemical reaction can be separated by, मतलब जो डीएनए फ्रैगमेंट हम जनरेट करते हैं with the help of restriction endonuclease, उसको हम सेपरेट कैसे करें? How we separate the fragments of DNA? So first option is the polymerase chain reaction. Next option B is the electrophoresis. Option C is the restriction mapping. Option D is the centrifugation technique. Please answer in the poll. I'm launching poll for you. Please answer first. You have 1 minute to solve this question. After 1 minute, we explain the question. Okay. Correct, JNV Visakhapatnam. Correct, JNV Dahod. So I am in this poll, and here is your answer. Most of people choose an option B, and obviously option B is the correct one. So they ask about the separation. So here the separation technique is there have two separation techniques here. You can see first is the electrophoresis technique, and next one is the centrifugation technique. But generally, centrifugation technique used for the cell contain separation. Cell contain separation. Okay. Or the centrifugation technique is used for the broad level of separation. But if you want to separate DNA molecule on base of their size, okay, you need to choose electrophoresis technique. Okay. So here you can see the assembly of electrophoresis technique. Okay. And here you can see this is the gel portion. Okay, this is the gel, agarose gel, we can say. These are the electrodes. Here the negative charge, here the positive charge. This positive charge electrode is known as the anode, because negative charge DNA, okay, negative charge DNA attract to the electrode. That's why it is the anode. So next, this is the buffer. Buffer is important to maintain pH इन दी प्रोसेस. मतलब यहाँ पर हम क्या करेंगे ना, वाटर को ऐड करेंगे, एंड दिस जेल सबमर्ज इन द वाटर, ये रहेगा पानी के अंदर. ठीक है. सो व्हेन वी हीट, और वी कैन से व्हेनेवर द वाटर कम्स in the charges, like negative charge, positive charge, or when it comes in the electrical field, यहाँ पर उसका pH चेंज होने का चांस रहता है. देयर हैव high chance to change pH of that specific water. That's why we added buffer here. Here you can see the wells. These are the wells. Wells are the we can say this type of things. ठीक है, मतलब एक गड्ढे जैसा होता है, like hole. Okay. And here we load the DNA sample. यहाँ पर हम क्या करेंगे, DNA sample को लोड करेंगे. and this portion, this part is known as the bands of DNA after the electrophoresis process. Electrophoresis is a very long process. It takes 4 to 6 hours for the separation of DNA molecules. Okay. Polymerase chain reaction is the nothing but the amplification of डीएनए. मतलब इसमें क्या होगा, डीएनए को बढ़ाया जाएगा, नंबर्स ऑफ डीएनए बढ़ाए जाएंगे. Next one is the restriction mapping. So restriction mapping is the technique which important to find out length of DNA. ठीक है. So उसे हम कहते हैं, और unknown length of DNA we can say that thing is known as the restriction mapping. And we also known as the centrifugation. Centrifugation is important for the separation on the base of density. ठीक है. So that's why option B is the correct one here. Going toward the next part, next question on your screen is, during the process of isolation of DNA, chilled ethanol. Why we use a chilled ethanol? And option A is a precipitation of DNA. Option B, break open the cell to release DNA. Option C, facilitated action of restriction enzyme. Option D, remove proteins such as a histone. I'm launching poll again for you. Please answer fast in a 1 minute, then we will discuss the question. चलो जल्दी जल्दी आंसर करो. Correct Visakhapatnam, Jainvi, correct Jainvi Dahood. Okay, so I am in this poll. Here maximum people choose option A, and option A is the correct option. Okay. So during the process of isolation of DNA, chilled ethanol is added and it is useful for the precipitation of DNA. Option B is the wrong. Break open the cell, release DNA. Actually, this happen. Breaking of cell happen by the different type of enzymes. Okay. Next, facilitated action of restriction enzyme. This is also not answer. Remove proteins such as histone is not answer. So you can see in this picture. So this process is known as the spooling. Okay, so spooling, in spooling you can see the aggregation of DNA threads. Okay, why they use a chilled ethanol? Do you know answer of it? We know ethanol is used for the precipitation of DNA, but why chilled only? Why not room temperature or hot ethanol? Anyone answer? Please answer in the chat box. किसी को पता है why chilled ethanol only? Chilled ethanol ही क्यों यूज करते हैं? Actually, ethanol is useful for the precipitation of DNA. That means वो DNA को की solubility को loss कर देता है ethanol. So ethanol have a property, and with the help of that property, the DNA lose the solubility. And why we use chilled? Chilled is useful for the we can say protect from the denaturation. मतलब denaturation से रोकने के लिए. That's why we use the chilled ethanol in the activity. Okay. So next question on your screen is, so in the figure, the diagrammatic representation of E. coli vector PBR322, which one given option correctly identify its certain component? And option 1, actually this is the option A, B, C and D. Okay. So option one, original restriction enzyme, ROP, reduce osmotic pressure, hint to eco-R1 selectable marker, AMPR, TTR, antibiotic resistant genes. Okay, and I'm launching poll for it. Please answer fast. चलो जल्दी जल्दी आंसर करो इसका. Okay, so I'm in this question, in this poll, and here is the answer. Maximum people choose an option D. Option D is the correct option. So, ORI side is important for the origin of replication. Origin of replication. Okay. Next, ROP is important for the replication process. That means ROP have a DNA, or we can say ROP is important to make some protein, which important for the replication process. Okay, replication process. Next one, hint to eco R1 are the restriction enzyme. ये क्या है? Restriction enzymes. Okay, and that's why the correct option here is the ampicillin resistant, tetracycline resistant, or the antibiotic resistant G. That's why option 4 is the correct one. Next, which one of following case? Which one of following is the case of wrong matching? Wrong matching किसका है यहाँ पर? Somatic hybridization, fusion of two diverse cell. Option B, vector DNA site for the tRNA synthesis. Option C, micropropagation in vitro production of plant in a large number. Option D, callus, unorganized mass of cell produced in the tissue culture. ठीक है, so I am launching poll for it. Please answer fast. You have 1 minute to solve this question. Correct, JNV Visakhapatnam. JNV Visakhapatnam, we have other people also solve the question. That's why we need to give time for them. So हम इतना जल्दी फास्ट नहीं कर सकते, ठीक है? हाँ, we like reduce the time, like 60 सेकंड का 40 सेकंड कर सकते हैं. So like अभी 40 सेकंड हो गया. I end this poll and this is your result. Maximum people choose an option B. बहुत ही easy question था. So question क्या है? Somatic hybridization. Hybridization, we know in case of somatic hybridization, there have fusion of two diverse cell. Vector DNA useful to transmit our we can say gene of interest in the cell. Okay, so it not a site for the tRNA synthesis. That's why option B is the correct option. Micropropagation, it is the in vitro production of plant in the large number. This is the correct. Callus is the unorganized mass. Okay, that's why option B is the correct one. ठीक है, चलो आगे बढ़ते हैं. Next question on your screen. Which one is a true statement regarding DNA polymerase used in the PCR? Option A, it is used to ligate introduced DNA in a recipient cell. Option B, it serves as a selectable marker. Option C, it isolates from the virus. Option D, it remains active at a high temperature. Okay, चलो. So I'm launching poll for it also. Please answer fast. And then we will go to the next question. People who join from the YouTube live, they can answer in the chat box of YouTube. Chat box YouTube का ओपन है, उसमें आंसर करो, ठीक है. Okay, I end this poll, and this is your result here. Some of people, like 33 people given answer B, and 67% of people choose an option D. So देखते हैं यहाँ पर क्या आंसर आएगा. 1 minute. हाँ. So question is, which one a true statement regarding DNA polymerase? They ask about the DNA polymerase use in the PCR. So we know when we use DNA polymerase in case of PCR, that DNA polymerase is the thermostable polymerase, and that polymerase is known as the tag polymerase. ठीक है, tag polymerase को हम use करते हैं, that come from the thermus aquaticus bacteria. Okay, so it is used to ligate into the DNA, not ligate नहीं करते. Ligate का मतलब होता है दो सेगमेंट को ज्वाइन करना, ठीक है. So we not join DNA. It serve as a selectable marker. No. Antibiotic resistant genes are used as a selectable marker. Next, it is isolate from virus. No, it not isolate from the virus. Option D is the correct one. It remain active in the high temperature because it have a ability of thermostability or stability, or we can say thermostable enzyme. Okay. So next question on your screen, please answer fast. Question is, for the transformation microparticle coated with DNA to be bombarded with the gun and are made up of, किससे बना हुआ है? Option A silver or platinum, option B platinum or zinc, option C silicon or platinum, option D gold and tungsten. I'm launching poll for it. Please answer fast. Then we go. Okay, so I end this poll. So here maximum people choose on option D. Option D is the correct option. Gold and tungsten are used in the gene gun. Okay, so you can see in this picture, this is the gene gun, and gene gun generally used for transport our gene of interest in the plant cell. Okay, so that's why option D is the correct one. Coming toward the next 1 minute. In genetic engineering, the antibiotic resistant gene is used to select healthy vector. Option B adds a sequence from where replication start to keep culture free of infection. Option D as a selectable marker. I'm launching poll for it. Please answer fast. तो जल्दी जल्दी आंसर करो भाई इसका. ठीक है, मैं पोल को एंड करता हूँ एंड हियर इज द आंसर मैक्सिमम इक्विवेलेंस इज एन ऑप्शन डी. ऑप्शन डी इज द करेक्ट वन. ठीक है, देखते हैं क्यों करेक्ट है. So in genetic engineering, the antibiotic resistant gene is used as a selectable marker with the help of antibiotic resistant gene. We can identify transform cell and a non-transform cell. ठीक है. So other options are the wrong, like it not useful for the create or select healthy vector. It not important for the as a sequence from a replication start, that are the right sequence. Next, to keep culture free infection, this is also not a correct option. Option D is the correct one, that is as a selectable marker. Okay, so moving toward the next. Here you can see. Okay, so here you can see ampicillin, tetracycline are the selectable marker site. Next, in figure show the 3-step ABC. नीचे एक फिगर दिया है जिसमें एबीसी हमें दिखेगा. दिस फिगर रिप्रेजेंट पॉलीमरेज चेन रिएक्शन. हमें ऑप्शन को चूज करना है, करेक्ट ऑप्शन को. एंड ऑप्शंस आर, ठीक है. सो ऑप्शन ए, डिनेचुरेशन ऑफ टेम्परेचर ऑफ अबाउट 50 डिग्री सेल्सियस. जो हमें ऑप्शन सॉरी 1, मतलब ये ए ऑप्शन रहेगा. ए पर हमें डिनेचुरेशन दिख रहा है 50 डिग्री सेल्सियस पर, दैट इज द ऑप्शन बी. यहाँ पर हमें सी, extension in the presence of each stable DNA polymerase. Option A, annealing with 2 set of primer. And next is the B, denaturation at temperature about 90 degrees Celsius. ये रहेगा D option, ये रहेगा C option. ठीक है, ये रहेगा D. चलो इसका आंसर करो. आई एम लॉन्चिंग पोल फॉर इट एंड आंसर फास्ट. चलो आंसर करो फटाफट. चलो मैं पोल को एंड करता हूँ. हमें कोई भी आंसर नहीं मिला पोल में, चलो कोई बात नहीं. सो यहाँ पर देखो, पहला जो स्टेप रहता है, जो फर्स्ट स्टेप इन द पॉलीमरेज चेन रिएक्शन इज द डिनेचुरेशन. ठीक है, मींस ये जो पहला प्रोसेस है, दिस इज द डिनेचुरेशन. एंड डिनेचुरेशन कितने डिग्री टेम्परेचर पर होता है? डिनेचुरेशन हैपन एट द 94 डिग्री सेल्सियस टेम्परेचर. देन नेक्स्ट प्रोसेस इज द एनेलिंग. मीन्स ये बी स्टेप जो दिया है, ये इसे हम कहेंगे एनेलिंग. सो व्हाट हैपन इन द एनेलिंग? डिनेचुरेशन में क्या होगा? द हाइड्रोजन बॉन्ड ब्रेक विद द हेल्प ऑफ टेम्परेचर. एनेलिंग में क्या होगा? प्राइमर विल ऐड. ठीक है, सो हियर यू कैन सी दीज आर द प्राइमर्स. ओके, सो यहाँ पर प्राइमर को ऐड किया जाएगा एंड दिस प्रोसेस हैपन ऑन द 52 डिग्री सेल्सियस. ठीक है, नेक्स्ट स्टेप इज द एक्सटेंशन प्रोसेस. ये रहेगा एक्सटेंशन. ओके, एंड दिस एक्सटेंशन हैपन ऑन द 72 डिग्री सेल्सियस. देन देखते पहला ऑप्शन, पहला जो ए ऑप्शन है उसमें ए में क्या बोला है यहाँ पर? डिनेचुरेशन. यस, डिनेचुरेशन हैपन हियर at the temperature about 50 degree. No, not on the 50 degree, because denaturation happen on the 94 degree Celsius. That's why this option is the wrong option. Next देखते हैं option B. So C, extension in the presence of heat stable DNA polymerase. Yes, in case of C हमें देखने को मिल रहा है, because extension, for the extension of DNA we need the polymerase. हमें किसकी जरूरत है? Polymerase. And we also know tag polymerase is a use for the extension process. So C option correct हो सकता है. ठीक है, चलो. So next, sorry, B option correct हो सकता है. That means C statement correct हो सकता है और step जो C step है वो correct हो सकता है. Next option is the C. क्या है? Annealing. So this A means denaturation process, not annealing. B is the annealing process. That's why this option also wrong. Next one is the B. B को क्या बोला गया? Denaturation. But B is the annealing process, and that's why this option also wrong. So denaturation at the temperature about 98. ये सारा चीज correct है. No issue. 94, 98 डिग्री ठीक है, चलो चल जाएगा, कोई बात नहीं, एक टाइम के लिए मान लेते हैं दिस स्टेटमेंट इज द करेक्ट, बट दे एडेड डीनेचुरेशन फॉर द बी पोर्शन, मतलब बी के लिए उन्होंने डीनेचुरेशन डाला, बट दिस इज द दैट इज बी पोर्शन नॉट डीनेचुरेशन. वो कौन सा प्रोसेस है? एनेलिंग प्रोसेस. एंड दैट्स व्हाई ऑप्शन सी is option C, in the sense option B is the correct option. That means step C is the correct step. Okay, and that's why here you can see the B, extension in the presence of heat stable DNA polymerase. ठीक है, hope you understand all the thing. चलो आगे बढ़ते हैं. Next question, which one of following represent palindromic sequence? So पहला 30 सेकंड उसको अच्छे से ऑब्जर्व करो, देन आई विल लॉन्च द पोल. ठीक है, पैलिंड्रोमिक सीक्वेंस पूछा गया है. ठीक है, चलो, सो आई एम लॉन्चिंग पोल नाउ, प्लीज आंसर फास्ट. चलो चलो चलो, बहुत इजी क्वेश्चन है. Correct, Jenny Dahoud. Okay, so I have aimed this poll, and this is your result. Maximum people choose an option D. Obviously, option D is the correct one. So I think you know about the palindromic sequence. Palindromic sequence के बारे में हमें पता है, उसके बारे में डिस्कस करने की जरूरत नहीं, सीधा सीधा देख लेते हैं. पहले पहले यहाँ पर CC दिख रहा है क्या आपके? CC है, यहाँ पर A, यहाँ पर T. So this is not a palindromic sequence. Next यहाँ पर CA हमें देखने को मिल रहा है. You can see CA here, again T here. You need to T here, but यहाँ पर T नहीं है. That's why this is also not a option, correct option. नेक्स्ट यहाँ पर G दिख रहा है, A दिख रहा है, चलो G दिख रहा है, A दिख रहा है. देन हियर यू कैन सी द T. यहाँ पर T दिख रहा है क्या? नो, ये इट नीड टू वी वांट T हियर. यहाँ पर हमें T की जरूरत है, बट T नहीं है, दैट्स व्हाई ये भी ऑप्शन नहीं रहेगा. हियर यू कैन सी GA देन AT देन AT. ठीक है, चलो हो गया, क्लियर है. दैट्स व्हाई ऑप्शन डी इज द करेक्ट ऑप्शन. चलो नेक्स्ट, ballistic gene gun is suitable for. Gene gun किसके लिए यूजफुल है? We already discussed in the previous question also about the gene gun. Next options are option A, transformation of plant cell. Option B, constructing recombinant DNA by the joining with a vector. Option C, DNA fingerprinting. Option D, disarming pathogen vector. I'm launching poll for it. Please answer fast. Then we will discuss the question. चलो जल्दी जल्दी आंसर दो इसका. करेक्ट, जेएनयू दाहोद. चलो हम पोल को एंड करते हैं. दिस इज योर रिजल्ट. मैक्सिमम पीपल चूज ऑन ऑप्शन ए. ऑब्वियसली ऑप्शन ए इज द करेक्ट वन. अब बैलिस्टिक जीन गन सुटेबल फॉर द प्लांट सेल. ठीक है, प्लांट सेल के लिए ये सुटेबल रहेगा. अदर ऑप्शंस आर द रॉन्ग. व्हाट इज द डिसआर्मिंग पैथोजन। डिसआर्मिंग पैथोजन का मतलब क्या होता है? सो वी यूज द रेट्रोवायरस viruses for the transport gene of interest. Means we use the retrovirus as a vector. But you know, retrovirus is the pathogenic, it caused the cancer or it caused the different type of diseases. So at that time, we use the insertional ability of virus. ठीक है, उसका जो insertional ability उसको ही हम यूज करेंगे, but we lost the pathogenicity. उसका जो पैथोजेनिसिटी है उसको हम लॉस कर देंगे, एंड दैट्स व्हाई हियर यू कैन सी द डिसआर्म। डिसआर्म मतलब उसके हाथ हम तोड़ देंगे, आर्म्स को उसके लूज कर देंगे। दैट आर्म्स मींस पैथोजेनिसिटी ऑफ दैट स्पेसिफिक वायरस, एंड दैट वायरस व्हिच हैव अ डिसआर्म, मतलब जो डिसआर्म है उनको हम क्या करेंगे, एज अ वेक्टर यूज कर सकते हैं। जनरली एनिमल सेल के लिए वी यूज द वायरसेस एज अ वेक्टर। ठीक है, चलो नेक्स्ट। गिवन बिलो इज अ सैंपल पोर्शन ऑफ डीएनए स्ट्रैंड गिविंग द बेस सीक्वेंस। 30 सेकंड का क्वेश्चन है, पढ़ लो आप ही और आप ही आंसर करो। ठीक है, सीधा सीधा क्वेश्चन है, ज्यादा इसमें झोलझाल नहीं है। ऑप्शन ए इज द डिलीटेड म्यूटेशन, ऑप्शन बी स्टार्ट कोडॉन एट 5 एंड, ऑप्शन सी पैलिंड्रोमिक सीक्वेंस ऑफ द बेस पेयर, ऑप्शन डी रेप्लिकेशन कंप्लीटेड। आई एम लॉन्चिंग पोल फॉर इट, प्लीज आंसर फास्ट। 30 सेकंड में आंसर करो, बहुत इजी क्वेश्चन। करेक्ट जैन विदाउट। Correct, JNV, Visakhapatnam. Chalo. So I end this poll. This is your result. Maximum people choose on option C, and option C is the correct one. So this is the palindromic sequence we already discussed in the last question. Chalo, next question. There is a restriction endonuclease called EcoR1. So we know the EcoR1. What does the co means? इसमें से co किसका नाम या किसे हम कहेंगे? Part stand for silom coenzyme coli colon. ठीक है, and I'm launching poll for it. चलो आंसर दो, जल्दी जल्दी दो। ठीक है, 40 सेकंड, 40 सेकंड ओनली। प्लीज आंसर इन 40 सेकंड। करेक्ट, जेएनवी, विशाखापट्टनम, दाहोद। You have 20 seconds to solve this question. Please answer fast. Okay, so I'm ending the poll. Here the answer. Most of people choose on option C, and option C is the correct option. Okay, so this means coli. So here you can see one of chart. This is the EcoR1. So first letter suggests genus, that is the capital letter. Next letter suggests species. Next, this is the species name actually. This letter suggests strain. Okay, R and 1. So 1 is the order. Okay, when we isolate the enzyme. Okay. So actually, EcoR1 is isolated from the Escherichia coli. Escherichia coli is one of bacteria, and that's why CO means the species name of that specific bacteria, and that's why option C is the correct option. चलो आगे बढ़ते हैं। Agarose extracted from seaweed find use in, ठीक है। अब अगैरोस का यूज किस में होता है? टिश्यू कल्चर, पीसीआर, जेल इलेक्ट्रोफोरेसिस, स्पेक्ट्रोफोटोमेट्री। ठीक है, आई एम लॉन्चिंग कॉल फॉर इट। चलो एक क्वेश्चन है मेरा, एनीवन नो अबाउट स्पेक्ट्रोफोटोमेट्री? स्पेक्ट्रोफोटोमेट्री किसी को पता है? ठीक है, विशाखापट्टनम, दाहोद। करेक्ट आंसर। We know the PCR, we know the electrophoresis technique. Anyone know spectro? Please answer me in the chat box. Anyone know about the spectrophotometer? ओके, चलो एंड दिस पॉइंट। सो यू नो द आंसर, उसके बारे में डिस्कस करने का बात ही नहीं है, बिकॉज वी नो द अगैरोज जो जेल रहता है, इट एक्सट्रैक्टेड फ्रॉम द सीवीड and it used in the gel electrophoresis process. So here you can see this is the gel, okay, जो electrophoresis process में use होता है. So I'm asking one of question you, that is a spectrophotometry. Spectrophotometry का मतलब क्या होता है? So whenever we like bombard the light, मतलब हम किसी सैंपल के ऊपर लाइट को बम्बार्ड कर रहे हैं. फॉर एग्जाम्पल दिस इज द सैंपल, इसके ऊपर हम लाइट को क्या कर रहे हैं, बम्बार्ड कर रहे हैं, ठीक है. एंड हियर इज द रिसीवर. सो हाउ मेनी अमाउंट ऑफ लाइट यहाँ से पास होता है, एंड हाउ मेनी अमाउंट ऑफ लाइट एब्सॉर्ब बाय द सैंपल, मतलब कितना अमाउंट ऑफ लाइट को एब्सॉर्ब कर रहा है और कितना पास कर रहा है, उसका जो कैलकुलेशन रहता है, इट हैपन इन द स्पेक्ट्रोफोटोमीटर. सो विद द हेल्प ऑफ स्पेक्ट्रोफोटोमीटर हम क्या चेक करेंगे, कॉन्सेंट्रेशन ऑफ द सॉल्यूशन, मतलब वो कॉन्सेंट्रेटेड है, डाइल्यूटेड है, ये सारी चीजें हमें पता चलती है विद द हेल्प ऑफ स्पेक्ट्रोफोटोमेट्री. ठीक है, चलो आई थिंक किसी ने आंसर भी किया मुझे, जेनवी दाहो. हाँ ठीक है, करेक्ट है. चलो आगे बढ़ते हैं इसी के साथ. नेक्स्ट क्वेश्चन, विच वन of following technique made it possible to genetically engineer living organism? This is also very easy question. First is hybridization, next is recombinant DNA technique, next is X-ray diffraction, next is heavier isotope labeling. ठीक है, चलो मैं फिर से पोल को लॉन्च करता हूँ. प्लीज आंसर द क्वेश्चन. ठीक है, इसका बहुत ही इजी आंसर है. ठीक है, सो मोस्ट ऑफ पीपल ऑप्शन बी को सिलेक्ट किया. ऑप्शन बी करेक्ट है. दैट इज रिकॉम्बिनेंट डीएनए टेक्नोलॉजी इज यूजफुल टू जेनेटिक इंजीनियर लिविंग ऑर्गेनिज्म, टू डेवलप जेनेटिक इंजीनियर लिविंग ऑर्गेनिज्म. अदर टेक्निक्स आर नॉट यूज फॉर दैट प्रोसेस. नेक्स्ट क्वेश्चन है, the linking of antibiotic resistant gene with the plasmid vector become possible with DNA ligase, endonucleases, DNA polymerase, exonucleases. I am launching poll for it also. Please answer fast. चलो आंसर करो फटाफट फटाफट, जल्दी जल्दी. Okay, so I am in this poll. And here is the answer. 50% people choose option A, 50% choose on option B. चलो इसका आंसर देखते क्या है. So they ask the linking of antibiotic resistant gene. मतलब हमारे पास क्या है, antibiotic resistant gene है. So we have a gene actually. हमारे पास क्या है, gene है actually. किसका gene है, antibiotic resistant gene. Okay, no issue. With the plasmid vector, तो हमें क्या करना है, plasmid vector में क्या करना है, antibiotic resistant gene को link करना है. The linking of antibiotic resistant gene with the plasmid vector become possible. अब लिंकिंग किससे करते, कौन सी चीज से हम लिंक करेंगे चीजों को. चलो ऑप्शन देखते हैं. डीएनए लाइगेज से हम लिंक कर सकते हैं, ठीक है, तो ये ऑप्शन हो सकता है, नो इश्यू. एंडोन्यूक्लियस से क्या करेंगे, लिंकिंग करते हैं क्या हम, नो, इससे हम लिंकिंग नहीं करते, उससे हम ब्रेकिंग करते हैं. पॉलीमरेज में क्या करते हैं हम, एम्प्लीफिकेशन करते हैं. ठीक है, नेक्स्ट वन एक्सोन्यूक्लियस आल्सो ब्रेकिंग के लिए यूज में आएगा. जो आउटर साइड का बेस रहेगा उसको ब्रेक करते हैं. एंडोन्यूक्लियस से जो इनसाइड बेस रहेगा उसको ब्रेक करते हैं, तो लिंकिंग करने के लिए हमारे पास कौन है, डीएनए लाइगेज। तो डीएनए लाइगेज इज द आंसर, ऑप्शन ए इज द आंसर। ठीक है, चलो मूविंग टुवर्ड द नेक्स्ट, दिस इज द लास्ट क्वेश्चन ऑफ आवर सेशन, today's session. Restriction endonucleases are enzyme which, restriction endonuclease हम किसके लिए यूज करते हैं, अगेन इट्स अ सिंपल क्वेश्चन। ऑप्शन ए है, दैट इज अ मेक कट अ स्पेसिफिक पोजीशन विद इन डीएनए मॉलिक्यूल। नेक्स्ट इज द ऑप्शन बी, रिकॉग्नाइज स्पेसिफिक न्यूक्लियोटाइड सीक्वेंस फॉर द बाइंडिंग ऑफ डीएनए लाइगेज। ऑप्शन सी है restriction the action of enzyme DNA polymerase. Next option D, that is the remove nucleotide from the end of DNA molecule. Sure. Correct, Jain in Visakhapatnam. Correct, Jain in Dahod. Okay, I am in this poll. This is your result. Maximum people choose an option A. Obviously, option A is the correct one. So they ask about the endonuclease. Endonuclease in between the gene, मतलब gene के बीच में से cut करेगा, ठीक है? So that's why option A is the correct one. Other option are the wrong. ये तो दोनों wrong है ही. But चलो, remove nucleus from the end of DNA बोला गया है. End of DNA molecule. That is the exonucleases. वो क्या रहते हैं, exonucleases रहेंगे. So that's why option D is not answer. Option A is the answer. So that is for today, guys. In next day, we will discuss about the question from this lesson. This lesson in the sense biotechnology first principle and processes lesson. Some of heavy questions in the next session. Okay. So thank you for joining. Goodbye to all.